Hi, everyone. Thank you for organizing this remarkable conference. I hope that everyone is well. I would like to share with you a few insights from our lab about the neural underpinning of empathy. And basically, I would like to argue for a paradigm shift in the field of empathy that views empathic interaction as a feedback loop between an empathizer and a target. First, I would like to thank my collaborators, the funding agencies, and the students that participated in running the experiments. There are many definitions to empathy, but it is widely agreed that empathy involves many sub-processes, some of which are more emotional, like emotional contagion, which is basically our ability or tendency to share our emotion with other people and feel other people's pain or distress. Other subcomponents are more cognitive, like perspective taking, which is basically our ability to cognitively understand the perspective of other people, their beliefs, their emotional state, sorry, without actual emotion sharing. Empathy occurs every day, all the time, in dyadic interaction, in group interaction, in positive emotion, in negative emotion, in adolescent, in childhood, across the lifespan. We see that empathy occurs every day, all the time. But it mainly involves this face-to-face dynamic interaction. And here is a short clip. I hope you can hear the sound. You can see a brother trying to console his younger sister. She's crying. He's trying to hug her. Then he tries another strategy and he tries to help her. And now she is happy. Now she's back to homeostasis. So we can see that this empathic reaction involves this feedback loop of trial and error. It tries one strategy, then he changes the strategy based on the feedback that he receives from her. And the question is, how can we examine this very complex behavior, this feedback loop, when we seek to examine the neural underpinning of this behavior? And unfortunately, most traditional approaches to study empathy are extremely limited. Most study in cognitive neuroscience involve isolated participant in a decontextualized environment with no real face-to-face interaction. And these conditions only remotely resemble the real face-to-face empathic interaction. And there are two fundamental limitations to the traditional approaches to study empathy. One is person-dependent and one is situation-dependent. The person-dependent limitation focuses on the limitation of our movement in basic scanning condition. When we limit the activity of the participant, we may basically interfere with the sense of embodiment and sense of agency. We know that embodiment is extremely important for our emotional experiences. And when we interfere with the sense of embodiment, we perhaps may interfere with their empathic reaction. The second limitation is situation dependent. When we have this artificial, out of context experiment, we don't really examine the situation of face-to-face interaction, and these interactions don't really simulate empathic interaction in real life. So the traditional approach suggested we should look at controlled experiment with controlled stimuli with no movement. And the new approach suggested by Leo Schilbach suggested we should examine two brain, measure brain and behavior in real face-to-face interaction. And the real-life extreme approach suggested that we should actually take our experiment outside the lab in real setting and examine dyad as well as a group in more natural situations. And in the context of empathy, when we study, have these controlled experiments, we basically look at empathy as an open loop. Information is transferred from the target to the empathizer with no feedback back to the target. However, when we use the two-brain approach, there is feedback from the target to the empathizer and back to the target. So we create those bidirectional feedback loop that allows a mutual alignment that can be developed between the empathizer and the target. And this mutual alignment can take the form of mimicry, movement synchrony, synchrony in facial expression, and also synchrony in brain signals. We recently developed a Tai Chi learning paradigm in the lab. We can see here the teacher, and here we can see the learner. The teacher is teaching Tai Chi movement to the learner. And we can see that very nicely their behavior becomes synchronized as they learn those Tai Chi movement. And we developed a way to calculate three-dimensional movement, three-dimensional synchrony movement in the lab. And importantly, we recently developed the one-way mirror setup that allows comparing between observational learning and interaction-based learning. In observational learning, there is a one-way mirror. The learner can see the teacher, but the teacher cannot see the learner. In the interaction-based learning condition, there is a transparent glass. The teacher can see the learner learner can see the teacher, and there are bidirectional signals between them. And we found that learning may be better in the transparent glass condition, and that the synchrony between the teacher and the learner is higher. We can see here, this is a zero lag correlation. We see that in the bidirectional condition, there is more synchrony between the teacher and the learner as compared to the unidirectional condition. So this suggests that it's in the context of empathy, it's extremely important to measure both the behavior and the brain signal of the empathizer and the target and examine how synchrony develops between them. So when we started to think about measuring empathic interaction and view empathy as a feedback loop, we found out this nice picture from the book of Franz de Waal. In this example that we see a juvenile chimpanzee putting his arm around an adult chimpanzee who was just defeated in a fight. Franz de Waal coined the term consolation to describe this tendency to reduce the distress using touch of target in distress. And he suggested that this is a very powerful strategy for distress reduction in chimpanzees, but it was also reported in bonobos, in elephants, and in many types of rodents. And of course, we see that across the animal kingdom, touch or empathic touch is used to reduce distress across human culture. Touch has been shown to communicate emotion. We can also identify emotion using touch. So we decided to focus on touch as our empathic response that can have a direct effect on the target. And when we looked for the type of touch that we would like to test, we first asked a group of participants to select out of 3 types of touch, which is the preferable touch for distress reduction. We presented 3 types of touch: slow stroking on the arm, which basically simulate the city fibers, which are known to be pleasurable touch fibers; slow stroking on the palm, which simulate the alpha beta fibers; and static hand holding. And what do you think that participants selected? Not surprisingly, 75% of our sample selected hand holding as the most preferable touch for pain reduction. So we decided to focus on hand holding And the first experiment carried out by Pavel Goldstein was a PhD in my lab and now has his own lab and will present later today. He sought to examine the analgesic effect of hand holding, and importantly, he sought to examine the moderating role of empathy on analgesia. So Pavel invited the 20 romantic couple to the lab, and he inflicted pain using a thermode attached to the arm to the female in 4 different conditions. He measured, the female was asked to rate the level of pain when she was alone in the room, when she was touched by a stranger, touched by her partner, and touched by, and not touched by a partner. Here, it was just mere presence of the partner. As we can clearly see from this figure, touch of the partner significantly reduced the level of pain of the target, suggesting that touch has an analgesic effect on pain. And importantly, we see that it is very important to also examine the identity of the toucher, because stranger touch had no effect on pain reduction. So this is very nice, although we know that touch is important for pain reduction. We use it all the time when we want to console children. But this is the first systematic evidence that hand holding actually reduces pain. But even more important, we looked at the partners, the toucher's level of empathy, and we start to examine whether it predicts the level of pain reduction. And as we can clearly see here, we found a significant correlation suggested that the level of empathy of the target significantly predicted the level of pain, such that empathic partners are basically better at reducing the pain of the target. Perhaps they produce a more attuned touch, perhaps they were more sensitive at sensing the touch of the pain of the target, but they basically produce a more effective touch, and they were more effective in reducing the pain of the target. In the second stage, we sought to examine the neural underpinning underlying this effect of the analgesic effect of touch using a dual EEG system. This study was also carried out by Pavel and in collaboration with Guillaume Dumas from the Pasteur Institute. And here again, we recruited 20 romantic couples. We collected we used the dual EEG system and focused on the alpha mu band, which was previously shown to underlie empathy, and it is also a biomarker of the observation execution system or the mirror neuron system. So we basically extracted the alpha rhythm from each participant and examined using a circular correlation, the number of correlation between different electrodes in different conditions. We had first a condition where there was no touch, no pain, condition with partner touch with no pain, condition with no touch pain, and the main interest condition was the partner touch pain. And each yellow line here represents a significant correlation between electrode. And as we can clearly see from this figure, the highest number of links between the brains, the highest level of interbrain coupling was found in the partner touch pain condition. So basically in the consolation condition where there was pain and hand holding, we see increased level of brain-to-brain coupling. And importantly, we found that the level of coupling predicted the level of empathy of the toucher, the one that was touching the partner. And importantly, the level of pain predicted, the level of brain-to-brain coupling predicted the level of pain of the target, such that higher level of synchrony, interbrain synchrony, or higher level of brain-to-brain coupling predict lower level of pain, suggesting that this coupling is associated with diminished level of pain. Perhaps it is responsible or a type of mechanism of pain reduction. In the second stage, we thought to examine the neural basis, anatomical basis of this effect using fMRI. This was carried out by Adi Koritsky, my former graduate student. In this experiment, again, we recruited 20 romantic couples. They were randomly divided into target and empathizer. And we had the 2x2 condition. In one condition, there was high physical pain, heat stimuli using a thermode attached to the leg. And in the low pain condition, there was a low heat, which was a control condition. So it was high versus low heat stimuli. And we had also a hand holding versus a rubber ball holding. So it was a 2x2 design, pain, no pain, hand holding or holding a rubber ball. Now, since we don't have a connected scanner, we applied the serial design where we basically scanned both the empathizer and the target serially in a counterbalanced manner. In some participants, we first scanned the empathizer while the target was outside the scanner. And in the second stage, the target was inside the scanner and the empathizer was outside the scanner. And in half of the participants, first the target was inside the scanner and the empathizer was outside the scanner. So this way allowed us to obtain scans of both the empathizer and the target, only there was no, the recording was not simultaneously. So it's not really hyperscanning. Now here we see that we clearly had a nice replication of our behavioral findings showing that hand holding reduces physical pain. We see it in the high heat stimuli, but also we had some effect in the no pain condition, suggesting that hand holding also reduces the minor pain experienced in the low heat condition. So basically we see that hand holding reduces pain. The neuroimaging analysis showed that when we created the contrast, where we compared the hand holding plus pain condition versus the all other controlled condition, we found a nice activation, shared activation between the target and the empathizer in the inferior parietal lobe. We can see here in blue activity in the IPL of the target. In red, we see activation in the empathizer, and in pink or magenta, we can see the shared activation. So doing hand holding plus pain condition versus other condition, we see very clear activation in the inferior parietal lobe, which is also part of the observation execution and mirror neuron system. When we extracted beta values from this region of interest, we found significant correlation in activation, showing that although the brain signal was not collected simultaneously for both partners, we see seemingly this synchronized activation between participants, showing that they showed similar activation in this condition. Now, the IPL, as I mentioned, is part of the observation execution system or mirror system, mirror neuron system, together with the inferior frontal gyrus and premotor cortex. We recently suggested that this system is a core network and part of the alignment system that includes also the reward system and the misalignment distress system, including the dorsal medial prefrontal cortex, dorsal anterior cingulate. And these networks are repeatedly shown to be activated during tasks that involve mutual alignment, like movement synchrony, like empathy, and also conformity. So in all levels of alignment, we see that these systems are repeatedly activated. The IPL was also found to be part of the Synchrony, movement synchrony system. So we were not surprised to see that during hand-holding in the empathic condition, we find activation in this system. In the second stage, we used the psycho-physiological interaction analysis, of PPI analysis, that allowed us to basically analyze the regions that were coupled in their activity with the IPL during the hand-holding plus touch condition. And we found that the IPL activity was coupled in its activity with the superior frontal gyrus, a region that was found before to be important for in emotion regulation. So we concluded that the model that can be suggested from this experiment is that perhaps shared activation, we can't say coupled activation because it was not simultaneously, but this shared activation in the IPL perhaps activate in the target activation in frontal region, which are important for emotion regulation. And this activation in the emotion regulation region is contribute to pain reduction. So the synchrony that we see in the dual EEG experiment and the coupled activity or the shared activity in the IPL suggests that perhaps the mechanism of pain reduction during hand-holding involve coupling in the observation execution system that then triggers activity in systems of emotion regulation. Now, there are still some open questions that remain. First, in this series of experiments, we focused on physical pain. And the question remains is what about emotional pain? And also what happens when the interaction partners are not romantic couples, but strangers? And what about other responses other than hand-holding? Because we use in real life many other strategies to reduce the distress of other people. So to address this issue, we are currently examining in our lab a new paradigm that we call the emotional sharing paradigm, ESP, in which target verbally shares with the empathizer a real-life distressful emotional event for 5 minutes. And in a control condition, the target verbally shares a neutral everyday event from his life. So we use this paradigm to measure both movement synchrony, facial expression synchrony, and brain-to-brain coupling. To examine movement synchrony, we use the motion energy analysis system developed by Fabian Ramsayer from Zurich. We basically measure we take the video and measure changes in pixels between the right and the left side of the screen. And we can see here that we found in our pilot study significant higher level of synchrony in the emotional sharing procedure versus the control condition. We are also analyzing face-to-face interaction and analyzing emotional expression synchrony using the iMotion platform that compares action units and how they synchronize with the changes in action unit in the partner. And our hypothesis is that basically synchrony in facial expression would predict distress reduction in the target. We're also measuring FNIR signal in this interaction and also predicted higher level of interbrain synchrony in the observation execution system would predict lower level of distress in the target. So basically, if until now, most models of empathy examine how a distress in the target affect the empathizer and which neural region participate in empathic response, we now suggest extended models of empathy that view empathy as a feedback loop. We see that the distress of the target triggers an empathic response. The empathic response may take various forms like hand-holding, mimicry, affect labeling, and in all these strategies affect back the target and can reduce the level of distress. And based on the gap that is identified between the emotion, the distress of the target and the level of emotion regulation needed, the response is adapted by the empathizer. So we can perhaps also adapt the models of reinforcement learning to see how the response of the empathizer change and adapt based on the responses of the target. Finally, before we conclude, as I mentioned in the beginning of my talk, empathy sometimes also involves group behavior and not only dyadic behavior. And we try to address this issue in the lab, and there are many examples of group synchrony in the animal kingdom. We see flocks of birds and schools of fish, and also we see flocking in humans. And we try to model it in the lab using a new paradigm that we developed in the lab, where we ask participants to move first randomly in the screen, and then they are required to move in synchrony. So they create like a flock of birds, they move together. And we found first that the participant with a high autistic trait perform poorly in this task as compared to participants with low autistic traits. We also use FNIRS in dyas and group to measure brain-to-brain coupling while performing the task. And initial evidence from our dual FNIR system shows increased coupling in the inferior frontal gyrus while performing the task. And we see this increase in coupling as they improve their performance during the synchrony task. And we seek to further examine it now with the group of 4 participants and see also how it is related to empathic behavior. In future study, we also plan to examine this empathy feedback loop with models of reinforcement learning and try to create paradigms that allow measuring how participants learn and adapt their empathic responses based on feedback. We also seek to examine individuals with psychopathology, for example, individuals with autism, with depression, with post-traumatic stress disorder, and other populations that are known to have difficulties in their social behavior, and to see how they how their empathic reaction in real face-to-face interaction is reduced. Again, I would like to thank my students, collaborators, and all the funding agency, and thank you for listening.